Okay, everybody, welcome to week 5 of BIO113. This week we're going to talk about cell replication and genetics. Uh, Basic overview is organisms inherit genetic information, and this information gets transmitted from the parents to the offspring via some specific regulated cell reproduction. Okay, so we know cells are, every living thing is made up of cells, and cells will replicate or make copies of themselves in a process called mitosis for your somatic cells. So for you, it would be every cell in your body except your reproductive cells. Those cells, the reproductive cells, undergo meiosis, which is a different process, very similar, but it's more complex, and we'll talk about that a little bit as we get into it. Just as a little bit of bonus info down here at the bottom, the billion plus cells of which you're made up, which you yourself have, usually on average every 7 days are regenerated. So you feel like a new person every week. Now, that being said, your brain cells do not regenerate. The only part of your brain that can make new cells is this little area called the hippocampus, where you have memory storage. Other than that, that's it for your neurons. Okay. So, mitotic cell cycle, mitosis. The cell cycle has 3 major phases. You got interphase, mitosis, and cytokinesis. And I tend to clump cytokinesis in with mitosis because it kind of happens at the end of mitosis, but almost simultaneously. So, and all cell cytokinesis means is cell division. So, interphase, this little cartoon over here, and shows you a nice picture of an entire cell cycle. And you can see a large portion of that is the interphase, where a lot of cell growth is going on, the DNA replication, and things like that. So you've got these 3 subphases of interphase called G1, S, and G2. G1, your cell's growing all the stuff inside of it, except the genetic information is duplicating and making everything, preparing for the cell to divide. And then S phase, you get your DNA gets duplicated by the cell. And then the G2 phase, it's kind of a checker on the S phase and everything. Make sure everything's ready to go. Make sure there's no mistakes in synthesis of the DNA. And if that's all good, then we proceed into the mitotic phase or mitosis. All right. So I mentioned this just a little bit a second ago. Regulation of this. There's a lot of, it's a very complex process, and there's a lot of regulation that goes on to make sure everything is good to go before we go to on through the cell cycle. So you've got a major checkpoint located in 3 spots in the cell cycle. One is between the G1 and the S phase. So G1, you get that cell growth going on, and before it wants to proceed to the DNA synthesis, it wants to make sure everything's in place. And if that's cool, then we go on to the S phase where we start the DNA synthesis. And then there's a checkpoint between the G2 phase and the M phase, the mitosis phase. And this is really just making sure it goes back and checks, is all the DNA synthesis correct? Are we ready to go into actual cell division? And if that checks out, you go into mitosis. And then also in the mitosis phase, there's a checkpoint during spindle attachment at a certain phase. We'll talk about, wants to know, are all the chromosomes attached? Can we separate these guys? Ready? Are they ready to go? So, in the complexity of the system, it has checkpoints to make sure everything's looking good throughout. Okay, so I'm going to show you, this is interphase, and this is an image of what that looks like. At the top here, I've got both a picture of from a microscope. On the left, we've got the cell. We've got a picture from the microscope, and we've got a cartoon. We got animal cells. And then on the right, we've got plant cells. We've got a picture from the microscope here and a cartoon, just so you can kind of see what each phase looks like. Now, interphase, inter means between divisions. So this is, like I said, this is the largest portion of the cell cycle. This is going to be where your cell gets everything ready, grows, there's DNA being replicated, and everything's getting ready to go for cell division to start. Okay, you get these specialized structures like centrioles formed, and you've got your DNA in here, and your DNA is kind of just loose, hanging out, you know, like it normally would be in the cell, very diffuse. You couldn't recognize a chromosome in here because you can't tell what a chromosome is. And you can't really see this much in the animal cell, but in the plant cell, you can see very diffuse looking nucleus. Okay. So we move into mitosis. And in mitosis, we've got 4 distinct phases of mitosis. We've got prophase. Pro means first, so this is the first phase of mitosis. And in this phase, the chromosomes start to condense and form very tightly wound structures, okay? They get really thick looking and they shorten up, and they're able to be seen under a microscope. And you can see here, much different from this picture where you've got, you know, a very fuzzy looking thing. You start to see actual chromosomes condensing and forming here. And once that does happen, you get this nuclear envelope, the membrane that surrounds the DNA, the nucleus, starts to dissolve. And these centrosomes start to make these mitotic spindles, these little protein microtubules are called. And these things end up connecting to all the chromosomes because so there's 2 of these, they go to opposite sides of the cell and they'll connect, each one of them connect to one side of each chromosome, okay? So then I wanted to show you how do you get from just DNA from the diffuse looking DNA to what you would see as far as a chromosome looks like. So you start out, this is what we're used to seeing for DNA. You got this double helix. So you've got, you know, your backbone, DNA backbone, and you got the base pairs connected here. They show them as a line, but there's 2 there connected. Well, this ends up coiling around. There's 8 of these proteins called histones, and it does a double coil around that. So that's one layer of condensing that the DNA will do. And so once it gets wrapped around these histones, they look like beads on a string, and you can see the picture here is what they look like. And then that actually ends up getting wound in a helix again. So it's tightly wound there. And then it goes on, that winds into a bigger helix. So it's very tightly wound. And once that happens, you see down here at the bottom, this is actual electron micrograph, whatever picture of what it actually looks like in real life if you were able to zoom in and look at a chromosome. You know, a lot of times we'll show, especially in this presentation, I'll show you cartoons of chromosomes. This is what a chromosome looks like, okay, condensed. It's joined at the center by this centrosome, centrosome, I think. Anyway, this during metaphase, this is in prophase, your chromosomes condense to look like this. So they start out really loose and they get really condensed down and tightly packed, okay? This information is just kind of, this won't be on the exam. I just want you to get an idea of what's going on here. All right, so the next phase of mitosis is metaphase. Meta means middle, and they use that term because here the mitotic spindles will, once they're attached to these chromosomes, they will line them up along a, we call a metaphase plate, kind of an equator like in the cell. So you see your centrosomes go to opposite ends of the cell, and they attach these spindles to the chromosomes and line them up. So that's metaphase, okay? Oh, and let me show you in the plant cell, you can see it very clearly. Here it is in the animal cell. It's kind of hard to see the actual chromosomes here, but in the plant cell, you can see the actual chromosomes lined up here along the metaphase plate. All right, next step, anaphase. Ana means to separate. And so we're actually, this is where the chromosomes, the duplicated chromosomes get separated from themselves. So their copies get separated into opposite sides of the cell. So they pull apart, physically pull apart. These microtubules will physically pull apart and push against each other to help separate the chromosomes themselves. You can see that going on here. They're kind of looking like they were, you know, fingers reaching out for each other. So the next step is telophase. Tele means end, and cytokinesis gets lumped in here together. So this top would be telophase. You've got your chromosomes all pulled to opposite ends, and you can see that in the animal cell as well as the plant cell. And as soon as that happens, you get this in plant cells, you get this cell wall forming to separate the two cells into two new cells. In animal cells, what you get is something called a cleavage furrow. And you can see in this image right here, it's this little contractile ring that gets smaller and smaller until the two separate. And at the same time this is happening, then the chromosomes get surrounded again by nuclear envelope forms right around the chromosomes, and the mitotic spindles dissolving, and then after all that's done, the chromosomes go back to uncoiled state, okay? So all this kind of happens very quickly. You've got chromosomes uncoiled to become chromatin again, the nuclear membrane reappears, you get the cell splitting in two, you get your cytoplasm gets evenly distributed, and the two cells separate from each other. And like I said, plant cells don't have a cleavage furrow. They use this cell wall to separate them, and it kind of forms in little bits and then joins together. Okay. So now that we've talked about the different phases of the cell cycle, we can take a look at a image of, this is an onion root tip. And we use the root tip because it's growing quickly and you get a nice cross section of cells in all different phases. So we slice this onion root tip and stain the DNA in here so you can see it very clearly. And you can see different phases here of the cell cycle. So here we've got a lot of cells that are in interphase. Remember, we've got, it's very loose looking and not condensed at all. So these would be interphase here, a lot of those. And then you've got prophase, the beginning, where you start to see the condensation of the chromosomes. You can see that here. You start to be able to see the chromosomes. And you get metaphase. They line up along the plate, like this one here, and this one here is really nice. And the next step is anaphase, where they start to pull apart. Here's a great example of that. Here's another example. And then telophase. I would say this one is close to telophase. This is definitely in telophase. You can see they're pulled apart. And once this happens, it moves right on into cytokinesis, where you get the cell wall formed. Let me see if I can find one here that is going. There's one on here I can tell somewhere that has that happening. I saw it earlier. We'll go with this one here. This is a good example. So you can see you start to get this plate between the two of them, and they start to dissolve, or not dissolve, but become less dense, and the nuclear membrane forms around them. This is a great one here too. It's another step to that where they just split and you're starting to form the cell wall, or you form the cell wall. Okay, so knowing all that, you can think of this onion root tip slice as being frozen in time, right? It's a picture of what's going on with cell division at any point in time. And because we've kind of frozen the cells in time, we can count how many cells are in each phase. And if you were to count, I want you to count 100 of these. I find this picture easier to use than the one that's in the actual question set, but either way, it's fine. Count as many as you can and get a total number of cells. Okay, so if I count the number that are in interphase, okay, I'm not going to count them right now, but if I counted them, let's say there's 100 cells here. There's not, but let's say there is. Let's say I counted 70 that were in interphase, and I counted, 10 in prophase, 10 in metaphase, 5 in anaphase, and 5 in telophase. Okay. Knowing that, I know that 70% of these cells are in interphase, so I can make an easy assumption here and say, okay, the cells are spending 70% of their time in interphase, right? Since this is a snapshot in time. And, it would be, if I said there was 10 in prophase, 10% of the time would be in prophase. So if I know how long it takes for the cell, for this onion root tip to do a complete cell cycle from beginning to end, and let's just say that is 10 hours. Okay, that's not really what it is, but we're going to say for purposes of this explanation, we're going to say, so if it takes 10 hours to do the whole cell cycle, and if 70% of the time is interphase, then I can tell you that it takes 7 hours to go through interphase. And if 10% of the time is in prophase, then I know it takes 1 hour for the cell to do prophase. So you can see how you can calculate how much time each phase takes. Now, the percentage of time, the ratio of cells that are interphase to cells that are in metaphase to prophase and all that, if you change the speed that this cell cycle is going to take, you're going to change, let's say you put this in a lower temperature, a little colder. It's going to slow down how long, it's going to make it longer for the cell to divide. But the percentage of cells in each phase is not going to change, okay? So while the overall time may change and the overall time for each cycle may change and be longer, the amount of cells in each cycle is not going to change. So the percent of cells in each cycle is going to stay the same. So if instead of 10 hours, it takes 20 hours to do a cell cycle, it's still going to be 70% of the cells are in interphase, but it's going to go from being 7 hours to 14 hours total time for that phase, okay? And that, I think we talk about that a little bit in the question set, and I wanted to make that clear to you, okay? Here's a nice video we're going to watch. Hopefully you can see this video. See if I can share this video with you. New share. Yes.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands known as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Okay, let me stop sharing that. That was a nice little video. And like I said, you can get to that in the notes. I'll post these slides every time, so you can always access the slides and get to that video. Okay. So moving on. So now that we talked about cell cycle for mitosis or interphase and mitosis, I want to talk a little bit about sexual reproduction. Okay, so mitosis is what your somatic cells undergo, right? And when you do mitosis, you want basically identical copies or nearly identical copies to be made. But when your sex cells, your gametes, divide, you want, it does a process called meiosis. And there it actually wants to promote some genetic diversity. And how does that happen? In a couple different, three different ways, basically. Well, two in the cell cycle, and then one we'll talk about. So in the prophase portion of meiosis, you actually get what's called crossover happening. And this happens quite a bit during this phase. You'll get, let's say you've made this, let's look at this chromosome here. You get the copy from your mother and the copy from your father. And this will actually, it will switch pieces, end pieces, we'll say, with each other. It's the same set of genes, but they're different versions maybe on each chromosome. And you can see a flip-flop going on here at an event called crossover. And this is promoted basically during meiosis to increase your diversity. So you can get, pieces from your mother's chromosome stuck on your father's chromosome and vice versa. That's one thing that happens during meiosis to promote this diversity. Another thing is you get this independent assortment. Okay, and what I mean by that is, let's say this cell we're looking at has 3 chromosomes. And when we make our copies, one side's going to have the mother's chromosomes and one side's going to have the father's. So we'll say the red's the mother and this one, and the blue's the father. When these things get lined up in metaphase, it doesn't really care which chromosomes on which side. It doesn't care if you've got, in this image, we've got 2 of the mothers on the top half and 1 of the fathers on the top half. And on the bottom half of the cell, you've got 2 of the fathers and 1 of the mothers. And it doesn't really care which way it goes here. It does this purposefully to get a nice mixture of your chromosomes. So, your parents, or let's say your sex cells, you have a gene from your mother and a gene from your father, or a chromosome from your mother and a chromosome from your father. When you make your sex cells divide, it'll split those into separate cells in the end, but it doesn't care if chromosome 1 is, from your mother and chromosome 2 from your mother, and chromosome 3 is from your father. It doesn't care. And does this purposefully to help kind of mix it up. Now, another way we, as animals, increase our genetic diversity is through mate selection. You choose different mates based on whatever you or whatever an animal sees as a quality, and that will add their genetic makeup to the offspring. Okay, so that is a way we increase genetic diversity on the cellular level, though, is these two sources for variation. All right, so here's an overview of meiosis. And basically, meiosis is the exact same as mitosis, except you do 2 rounds of cell division, okay? So you have prophase 1, just like prophase in mitosis. Metaphase, same thing, they line up along the plate. Anaphase, they separate, and telophase, they make 2 new cells. If you stopped here, it would be very similar to mitosis, except for this added crossover that you can see, and this independent assortment. You get, well, this happens in your cells as well, in your mitosis as well, but in meiosis, it definitely increases your diversity. So once you get these 2 new cells that have a normal number of cells in them, or of chromosomes in them, those will split again as well. So you get those 2 cells come down here and do another round of cell division, you get another metaphase, another anaphase, and another telophase. So in the end, when you start with 1 cell, you end up with 4 cells. And the reason you do this is in this last set of cells, these are your reproductive cells. You want to have half as much genetic information as you would in a regular cell, because what happens is egg and sperm join together, each bring in their own half number of chromosomes, so that they together form a complete cell that has a full complement of chromosomes. So that's why in meiosis, you actually do 2 rounds of cell division, okay? And this is the same thing again. Meiosis produces your gametes, okay, your eggs and your sperm, and you end up decreasing the number of chromosomes by half. So you end up with what's called haploid cells. Normal cells are diploid, meaning they have 2 copies of each chromosome. Sex cells are haploid, 1 copy of each chromosome. And again, you get this genetic diversity by crossover events. As you can see here, you got a little red goes onto the blue chromosome, a little blue goes onto the red, and by this independent assortment into different gametes down here, okay? Now, human chromosomes. We, as humans, have 23 pairs of chromosomes. 22 pairs are what we call autosomes, and then we have 1 pair of sex chromosomes. As you know, you're either XX or XY. XX means 2 copies of the X chromosome, and XY means a copy of the X and a copy of the Y. If you're XY, you're male. If you're XX, you're female. When you see this image of all the chromosomes laid out and paired up, this is called a karyotype. And they use this in genetic diagnosis in medicine to look at the chromosomes and make sure everything looks good and everything. Now, I said we have 23 pairs of chromosomes. The number of chromosomes doesn't necessarily indicate how complex a living organism is. Rats have 92 chromosomes. Ferns have 1,200 chromosomes, which is kind of crazy to think. And they're very low complexity compared to humans or animals in general. Mosquitoes got 6 chromosomes, and that's all he needs to bite you and suck your blood. Okay? All right, so let's talk about genetics, some basic principles of inheritance. Okay, so You may or may not have heard of this guy, Gregor Mendel, studied pea plants in the 1840s, a long time ago, before anybody knew what DNA was or genes or any of that. This guy figured out on his own, based on observable traits, things he could see with his eyes. He could see flower color or the color of the peas or the smoothness of the peas and things like that, and figured out by making crosses and keeping detailed records that these traits were things that were seen or were carried through the gametes or the sex cells, in his case, pollen, and he could see that these things contributed to the phenotype, depending on which ones got expressed or where he could tell the difference. And you had to have two parents, and each one contributed one form of a gene called an allele. And so you could have, they could each have different alleles, and you could see which one was expressed. So he did a lot of research and came up with two laws. One of them is the law of segregation, meaning when the gametes are formed, each version of a trait, an allele, separates from each other so that only one allele from each pair is contained in the gamete. And that's what we talked about during meiosis, is that you only have one copy of genetic information in each cell, sperm or egg. And this is known as law of segregation. So he figured this out without even knowing what chromosomes were or any of that. He figured out the law of segregation. And then he also figured out independent assortment. So during this gamete formation, during the pollen formation, each pair of these alleles got randomly assorted into the gametes. And he figured this out by doing all these crosses, and he could see that it was kind of random as to which pollen it went into, which gamete it went into. So this became known as the law of independent assortment. All right, so let's talk about these alleles a little bit more. So the things you can see, the observable, measurable characteristics of a gene are its appearance, like blonde hair, how tall someone is, things like that. These are referred to as your phenotype, okay? The things that you can see is the phenotype. What makes up this phenotype, the genetic makeup, or the genes that are responsible for this expression, this phenotype, is called a genotype, okay? So you have one gene from your mother and one from your father. So if you're talking about eye color, for instance, let's say your mother has blue eyes and your father has brown eyes. You'll get, a gene for blue eyes from your mother and a gene for brown eyes from your father. Now, one of those is going to be expressed, not both. And that's where we get, so the blue and the brown gene are alleles of each other. They're alternate versions of the same gene, okay? And they can produce these phenotypes that we see. So here's an example. We got heterozygous. That means the two alleles are different. So you've got, in this example, we say tall is the gene we're going to look at, the tall gene. And we give tallness a capital T and shortness a lowercase t. So if you have one copy of the tall gene and one copy of the short gene, you've got two alleles that are different. So we call that heterozygous. So you can see here the capital T and the lowercase t. Now you can be homozygous, where the two alleles are the same for that gene. So you can have two copies of the tall gene or two copies of the short gene. You would be homozygous. If you have two copies of the tall gene, you're homozygous for the tall gene. Two copies of the short gene, you're homozygous for the short gene. Now, if your two alleles are heterozygous, meaning you have one copy of the tall gene here and one copy of the short gene, the dominant allele, which we give the uppercase letter in this case, is the one that gets expressed over the recessive allele. So you don't see the recessive allele, you will see the phenotype will be the dominant allele. And the reason that is, I couldn't tell you why one gene's dominant, one thing's dominant over the other, but these are things that can be measured. And let's just say we'll go back to the eye color example. If blue is the dominant and you have one copy of the blue gene, the blue allele, and one of the brown alleles, if blue's dominant over brown, you're just going to see blue. You're not going to see the brown. If you're homozygous for the blue gene, you have two copies of the blue gene, blue genes, the blue eye color gene, you're definitely going to have blue eyes. The only time you're going to have brown eyes is when you have two copies of the recessive allele, the brown color allele, right? So if you're homozygous dominant, you're going to have the dominant allele. If you're heterozygous, you're going to have the dominant allele being expressed. And if you're homozygous recessive, then it's the only time you're going to see the recessive allele, okay? That's all I have for the presentation today. I am going to have in the folder, obviously, we're going to have a question set that is about a lot of stuff we talked about, covers cell cycle, covers this onion root tip thing. And then there's going to be a quiz, and then there's going to be 3 Labsters this week, 3. Okay, there's one on meiosis, there's one on mitosis, and there's one on genetics. So you got to do all 3 of those. All right, I'll give you more time because to do all 3 of those, because those things are so much fun. And of course, if you have any questions, just send me an email. All right, have a good week.